Hey, it's a science night. Okay, it says that we're live. Now I will wait for a cue from everyone to let us know that yes, indeed, they can see us and hear us and we are live and that everything can start. And then I'll do the starting, and we like starting. Hello, everyone. Are you there? I see one. There it is. We have the cue to begin in 3, 2. This is TWIS. This Week in Science, episode number 762, recorded on Wednesday, February 26th, 2020. To MicroBE or not to be? Hi, I'm Dr. Kiki, and tonight on This Week in Science, we will fill your head with moons, mysteries, and microbes. But first, thanks to the Burroughs Welcome Fund and our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. When James Watson and Francis Crick gained fame for discovering the structure of DNA, Erwin Chargaff said of them, without naming names, that in our days such pygmies throw such giant shadows only shows how late in the day it has become. Literally throwing shade. Chargaff was referring to the fact that despite getting all of the glory, the heavy lifting had already been done by many others. Chargaff himself had discovered the symmetry that A's go with T's, G's go with C's. Rosalind Franklin's X-ray crystallography revealed the shape of the helix. And it was Oswald Avery who had initially figured out that DNA was where the genetic code was hiding in the first place. And there are many, many others who did the hard work, spent tireless days in the lab, sleepless nights thinking how they might crack the code of life itself. And along come two guys who have contributed zilch to nothing and nada, and yet somehow walk away with all the credit. But they were good at one thing, at talking to people and building cardboard models of other people's research, which eventually allowed them to publish a post-it note of a research paper, more of an outline really, a quick one-pager, a summary of the work of others on the structure of DNA. And while there were others who should have gotten more credit than they did, this is exactly what the world of science needs. The connecting of information, the unsiloing of ideas. Franklin and Chargaff meeting at the AAAS meeting or reading about each other's research online may have changed history a bit. The speed at which science answers is only ever limited to how many people know its questions. That and This Week in Science coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again to talk about science because that's what we like to do here. And we have a guest tonight, which is very exciting. We're going to talk about little bugs, little things, little life, little lives.
Yes, bugs in the largest air quotes possible. Bugs.
Microbugs. There we go. Yes. All right. I have stories this evening about the moon. I really just want to talk about the moon and maybe some old algae. Justin, what did you bring?
I've got another mystery archaic human that is maybe the oldest example of isolated reproduction within the hominin family history, as well as a multicellular life form without mitochondria.
This sounds all very exciting. We've got no man is an island and some little things down don't need energy. In the way
And
we
I
like to
also
think
brought
of
the billion-year-old seaweed, which means it's that it must be that amazing of a story this week.
Old algae. We love it.
Yeah.
Blair, what is in the animal corner?
I have greedy seagulls. I have stabby shrimps. And
Stabby.
I have a teeny tiny story about robot fingers.
Teeny tiny. Okay. I'll take your robot fingers and I will raise you robot poems.
What?
Oh, I can't wait for that.
I'm going to double down at the end of the show. That's right.
my gosh.
Show me your hand. Okay. We're going to talk about the science right now. It's time. I hope everyone's excited because I ate chocolate, so I'm all sugar and caffeinified and chocolatified. As we jump into the show here, I want to remind you that subscribing to TWIS, the TWIS podcast, on your favorite podcast platform is fairly easy. You can also subscribe to YouTube or Facebook, and it'll bring you TWIS each and every time a new episode is published. Just search for This Week in Science or visit twis.org. Now we'll get into our science, and I would love to introduce our guest for the evening, the esteemed, the heroic Dr. Jonathan Eisen. Dr. Eisen is a professor at UC Davis in the College of Biological Sciences. He's the director of the UC Davis Microbiome Special Research Program and is affiliated with the Department of Evolution and Ecology, the Department of Medical Microbiology and Immunology, and the Genome Center. He's in his Genome Center office right now. His current research focuses on the evolution, ecology, and function of communities of microbes and how the microbes interact with each other and their host vessels.
Yeah, the hosts are boring, but they're there.
They're there. There's some interactions, the
Yeah,
microbes. That's really where the action is taking place. Yeah. Thanks for joining us tonight.
Very happy to be here.
Yes, we have been talking for some time about getting you on the show. And as I was looking at things that you're doing and thinking about things to ask you, I was like, I'm going to need several episodes of TWIS, and I really should have done this sooner.
Yeah, well, I'm here. I can come back.
Awesome. Perfect. Can you give us some background on how you came to be a microbial geneticist, how you came to be working in this area of research at UC Davis?
Yeah, I mean, there are two parts to this story that I will tell you. One is I discovered only after I became a microbiologist. I was going through papers that my mom wanted me to pick up from her house to get rid of. And I found a 9th grade high school essay where you could, for English class, where you could invent a question and answer it for yourself. And the question I asked for myself in 9th grade English was describe a step in the evolution of a bacterium.
Wow.
I don't remember being interested in this. I was an East Asian studies major for 2 years in college. I don't remember this at all. But then I switched to biology, and I know literally the moment I got into microbiology research was a visiting professor told me about this new professor that was about to start work at my college. And she worked on this amazing ecosystem in the bottom of the ocean. And she in particular was interested in these giant tube worms in the bottom of the ocean that have no mouth, no digestive system. They're very weird. They, are animals, but they don't function like animals. And they live off of bacteria that are inside their special trophosome tissue. And the bacteria take the energy of hydrogen sulfide and use it to fix carbon like plants' chloroplasts do for them. And I was just, this was amazing. Like, it was cool. It was in a weird environment. It's in the bottom of the ocean. It involves animals that function like plants. It involved microbes that they didn't at the time know anything about because they couldn't grow them in the laboratory. And it was right when DNA sequencing was starting to be used to study microbes. So I joined her lab as an undergraduate, and then I worked in her lab as a technician after I graduated. And then I was all microbes all the time, basically, from then on.
That's amazing. You've never looked back.
Not really, no.
Thank goodness. You're doing such interesting research. Tell us a little bit about the different projects that your lab is working on, because they seem, aside from being microbially oriented, they seem so disparate, so different in nature.
Well, I mean, part of my problem, and I think my success, is that I like everything. So I do work on lots of different things, but in part I do that because I'm a comparative biologist. I'm not interested in how one organism works or one system. I'm interested in what are the rules across lots of different systems. And in particular, for many years, so basically I mean, what I'm scientifically interested in is how new functions originate. And for many years, I studied this inside single microbes and how they invented new functions by mutating their DNA and doing various things to their genome. And in particular, I was interested in why different organisms had different rates by which they invented new functions, and something you can sort of call evolvability. And I did this, I studied DNA repair processes, and I studied how organisms regulated their mutation rates, and why, like HIV had such a high mutation rate and why some other organisms had a low mutation rate, and it affected their evolvability. Mm-hmm. And I did this in microbes, and I was very interested in this for a while. But what I changed to doing, and what I've been doing for the last 15 or 20 years, is really not how do organisms invent new functions from the inside, but how do they acquire new functions from the outside, either by stealing DNA, lateral gene transfer or recombination, or mostly how they interact with other organisms through mutualistic interactions or parasitic interactions or other types of interactions to invent new functions. And that's what I work on. And the problem and the advantage is I like every system. so I mean, someone comes to me and says, hey, here's this cool system. Are you interested in working on it? And pretty much the answer is yes. I mean, it just doesn't, and that's my, challenge. And I think, we've been successful in part by trying to compare all these different systems to each other. So right now, what I care about is how communities of microbes, collections of microbes on your skin or in your gut or on a plant leaf or on a tube worm in the bottom of the ocean or in a caterpillar or wherever, how they interact with each other and with their vessel, their host, to allow that system to colonize a new environment or degrade a new compound or come up with a new behavior and I want to know what the rules are. Like, what are the rules by which these interactions happen? So I need to do this in every system, in lots of different systems, and compare them to each other. So yes, you're right that it's hard to make sense of this. If you just look at the list of things we do, we work on corn and seagrass and rice and frogs and koalas and humans and the bottom of the ocean, and I could go on and on.
Have you talked about looking for, these rules, things that are common. Have you come across anything that seems to be standard across microbial communities? I mean, other than, hey, I can steal your DNA and be better than I was before.
Yeah, no, I mean, I think that the part of the searching for rules has led us to try to understand in particular. So for many years, I worked on single microbes that had a really big effect on their host biology, like a pathogen that kills the host. And therefore that phenotype is pretty strong. You're either dead or you're not. Or beneficial symbionts that the host can't live without, like the bottom of the ocean, the tube worm story. The worms need those bacteria to make their food and to make their vitamins and cofactors. And what I've gotten interested in and what I'm still searching for the rules for is, how does the interaction work when there are 10,000 species of microbes? And each of those species comes in 1,000 different forms. And, one host, it seems impossible for one host to co-evolve tightly with 10,000 species, each with 100 strains or 1,000 different strains. And so
Yeah.
I think the rules for these are microbiomes. The community of microbes is also known as a microbiome. And I think the rules are that hosts don't tightly co-evolve with single species. What the hosts do to interact with microbiomes is they set up the right niche, or they regulate whole clades of microbes. So they regulate all of the gram-positive microbes at one time, or all of the photosynthetic microbes at one time, or all of the things that crawl into their mucus layers at one time, because there's no way that they have really tight evolutionary interactions with all of those species at the same time. So they have to have interactions that are based upon bigger picture things, either phylogeny or function. And that to me is what we're trying to figure out, is like how, so like we do a lot of work on seagrass right now, marine flowering plants that live submerged in the oceans. They're very strange in the world of flowering plants. And just like with terrestrial plants, they have microbes that are associated with them. And what we're trying to understand is like, how can they recruit the right microbes to their roots and to their leaves and to their stems and to their flowers, and what are they secreting to do this? You know, what niche are they setting up? Are they feeding these microbes? Are they killing the wrong microbes with antimicrobial compounds? And those things seem to be the general patterns by which organisms interact with their microbiome.
Yeah. Or do they recruit the bacteria that will kill the other bacteria that they don't want around?
Yeah, I mean, so you know, I think anything's possible. I mean, I think the challenge is that communities of microbes are there whether they have a tight interaction with the host or not. And you'll see microbes in, you know, some place, and then the question is, what, if anything, are they doing? Do they have a function that is important for the host? And if they have a function, how important is that function? The more important the function is, the more likely it is that they have been co-evolving in some way, that the selective pressure is important there. So if they're just there and they don't provide any, you know, negative or positive effect on the host, they're likely not, you know, co-evolving in significant ways with the host.
Right.
And it's interesting. It's just the way you're putting this. And now I'm not thinking of them so much. The possibility is not having co-evolved, but the reason the seagrass is doing well in this area, or the reason an invasive plant does extraordinarily well in one location, might be that it happened to arrive in a place where the microbes are friendly for it, and not the other way around.
Yeah, I mean, that was actually our original question in getting at seagrass. I teach a very large introductory biology class here at UC Davis, and one of the other instructors was teaching the plant part of the course, and he was talking about the evolution of seagrasses, how 3 separate times aquatic plants invaded the marine environment and converged upon the seagrass phenotype. And they're related to each other, but they're in 3 separate evolutionary events. And I immediately asked the question that you're asking. I was like, did they bring their microbes with them, or did they recruit or get new microbes when they came to this place? And that's basically what we're trying to figure out now, is how much are their microbes terrestrial plant microbes that then evolved to tolerate the marine environment, or how much are they just marine microbes that were just there, and they are now colonizing the seagrass and doing something in association with the seagrass.
So it's definitely both, right? It's a question of how much of each. I don't know. I'm just, if I can draw an analogy, I'm thinking about red pandas and giant pandas, and the fact that their microbiome, they both share a lot of their microbiome because they both eat bamboo, but they're not very closely related. Yeah. But also, giant pandas share a bunch of their microbiome as well with other bears that eat meat, and they don't. So it's a both.
And the same is true like with all the human microbiome stories. That is, the communities are very complex. And if you say what is the pattern in the community, it could be 100 species have pattern number 1, like the red panda and the regular panda have shared community because of what they eat. But there's another 5,000 species there, of which some of them are shared among bears, and the bears have a particular microbiome, and some of them are shared among things in China, and some of them are shared among things that, lick the ground in some way.
Totally.
So it's, all of those things mixing together. And what's challenging and fun is that, we don't know in advance. I mean, when you first go to work on something, you don't even know if the microbes are doing anything interesting. I mean, they're certainly there, but you don't really know how much they're contributing to the biology of the system.
Right, whether they're just living there or whether they're
like I watched Toby
making
Hammer's,
B vitamins.
I watched Toby Hammer's interview with you guys on the caterpillar gut microbiome, and we just covered his paper on that in a class that I teach. And that's like a really interesting case study where there are microbes there, but there seem to be just transient vagrants that come through with the food. And they're, in some of the caterpillars, they're just not doing biology that is particularly important. And when you go to a new system, you have no idea what it's going to be.
Right, and it could be so many things. I'm really, for the first time, I'm really thinking of, in your website and stuff, you talk about the host is kind of a vessel, but it maybe more than that. It's the host is an ecosystem, and so the host could be a rainforest or a savanna or a tundra. And what do these different ecosystems support, and how do the microbes then come in, and which microbe species tend to come in and inhabit a particular ecosystem type?
Yeah, and actually one of the things that we worked on quite a bit for many of the systems is that first question, which is, if you go to a particular ecosystem, it could be a host or it could be a savanna or it could be the bottom of the ocean, and you look at the microbes there, what are the sources? Where did they come from? Yeah. So like in the human gut, people ask this a lot. Not just us. This is a common question, like, how many of them came from your mother during vaginal birth? And how many came from your home when you were growing up? And how many came from your pets? And how many come from your food? And how many come from your water? And how many come from
the sandbox where you played?
The toxoplasma that is in your sandbox. I'm sorry. And all of those things can be very informative because, the more regular that process is of succession in the microbiome in a particular system, the sort of easier it is to start to tease apart what's going on. And in some systems, it follows a pretty regular pattern, and in other systems, it's pretty chaotic. So it doesn't mean you can't study those other systems. It's just a very different approach to studying systems where the colonization of a niche by microbes is like random, as opposed to it follows some temporal pattern.
Right.
Yeah, because if you have the sort of blank biome there, and you put in a random selection of critters, you would expect them to sort of fill, like one of them at some point becomes more of a apex predator, one of them starts feeding on carcasses, or one of them starts doing this, that, or that. Right? They have this whole. They sort of diversify and find the available free resource niches to fill in. But with this massive population like this, and as diverse as it can be from one place to another, it sounds like you're saying that they don't always fill all of those niches in a way that you could compare it to the other one, or they are comparing to the others.
No, I don't think. I don't think they do fill all the niches, and I'm definitely of the camp that taking the anti-adaptationist point of view for microbes, which is, the adaptationist point of view that people have applied to single organisms, is basically you observe something and the probability is that it is beneficial. And if you can just think hard enough, you can figure out the likely benefit that thing is providing. And this is what, people call just so stories and other things. And, a lot of things obviously are adaptive, but many are not. And just as you see that with single organisms, you see the same thing with communities of microbes. There are people out there, I blog about this a lot. I give out an Overselling the Microbiome award on my blog, because there are a lot of people who think that all microbiomes are good. They're all good for the host, and they all are optimized, getting to your question, sort of, and they're optimized, and they're like this Gaia-like perfect ecosystem where everything works together and everybody's happy.
I think people with irritable bowel syndrome will argue that point.
I know, but the scary thing is that many people are arguing that all you have to do is treat those people with your favorite pill, and it will magically make their microbiome back to perfect, when there is no perfect.
Which I actually agree we will. I think we will actually get there at some point, but it will be a microphage. You just have to eat all this stuff away that you don't want to be there, while leaving the things that you consider beneficial. We'll get a little closer anyway.
Is this in a week or like 200 years?
No. This
Maybe
is
50?
we can figure this out.
Yeah,
we can figure this out. Or we can say we figured it out and make a lot of money selling.
Do you take probiotics?
I was just going to ask if you've tooled with your microbiome at all. So this is related.
So that's I consume a lot of prebiotic-like things. So the distinction is basically probiotics are living organisms that are supposedly beneficial, and prebiotics are the food for those probiotic organisms. And so there's clinical data that shows that some probiotics can have benefits to a certain fraction of the population for some health issues. Usually they're very subtle. They're like protection from infection in some cases after you've had antibiotics, or reducing inflammation. But there are some really good, well-studied probiotics for which there's clinical data that says that on average, they can help people. It's just on average, of course, they don't help every person. But most of the things called probiotics out there, or marketed as probiotics, there is literally no published data supporting the claims that they are making. And there is rational reasons to be skeptical of many of the claims that they are making. So I have taken some of the better studied probiotics occasionally when I've gone on antibiotics for a while and other things. I'm much more in the camp of trying to feed the microbiome smartly, rather than trying to. I mean, most probiotics also die very quickly after they're added to your system. That's of benefit to the people who sell them.
Right, you have to take more. Yes.
Yeah. But most of them are not, particularly adapted to the ecosystem that you're trying to shove them into. And they don't last for a long period of time. They still might have benefits. So some of them still are anti-inflammatory in some cases, but you need to take it again and again and again. And it's whereas if you modify your diet, you have ways of regulating the system at a bigger level, probably. But it's very complicated. I mean, the science is still in its infancy for figuring out what to do other than the traditional nutritional advice that we had even before we thought about the microbiome completely. Eat fiber, eat certain types of fiber, be smart about your diet. It's not that different in terms of the science, and there are a lot of people claiming otherwise, but most of the claims out there that they can tell you what to eat based upon your microbiome are, if they're crap.
So
fiber.
But this is also interesting because the insight that you have on this is that you are studying the microbiomes on place and maybe on a plant that is much easier to access than the internal workings of a gut and all the human interaction stuff that goes on with it. So the level of complexity here, you're saying, a little bit higher than maybe people are assuming.
Yeah, I mean, that's part of it. So studying poop in the thought that you're actually studying what's going on in the gut is a little complicated. I mean, it does work at some level, but it's imperfect. But in addition, you know, the bigger challenge, like if you compare animal models, where lots of people do microbiome studies in mouse and rat and other animal models, to humans, is mostly, to me, an issue of complexity. Like the animal models, they're kept in very controlled environments. They're fed very limited range of foods. They get, you know, interact with a very small population of individuals. They don't get exposed to the type of toxins and airborne other things that we get exposed to in normal life. And, you know, work on those microbiomes, it has been challenging to extend work on those systems to humans because of all the, like, each individual human has so much in their immune history and in their diet variation and in their interaction variation that trying to predict what's going to happen if you pop a probiotic pill is pretty hard for humans compared to laboratory mice.
I think it's interesting how much we've kind of swung from all bacteria is bad, just put antibacterial on everything. Yeah. And now we're like, ooh, bacteria is good. Microbiome, probiotics.
So it's interesting. I mean, I think that it's bipolar, actually. I think you still have both now.
Yeah.
So you have the camp that is saying all microbes are bad. And we still put antibiotics and antimicrobials in clothing, in shoes, in paints, in toothpaste, in cosmetics, in everything. And they are advertised and marketed as though they are going to protect us from all the microbes. Some of those same companies are now marketing microbiome safe products. So they're both saying we should kill all the microbes.
and save all the microbes.
In fact, take more microbes.
Yeah. It's like the star-bellied sneeches where you just you add microbes and you kill them, and you add microbes and you kill them, and it is amazing. And this is, again, I mean, I think a really important area of microbiology communication is it's clearly should be somewhere in between.
Yeah.
It's all gray. We talk about other jobs.
Yeah. But there are microbes that are bad, right? That will kill you. And the people that go around saying we should lick the subway poles in New York to boost our microbiome, they do say this. They should be taken away. And
I mean, they will if they continue to lick this.
Don't do that.
I've gotten into this conversation once before with my dentist, who was informing me in a very heavy tone that I have bacteria in my mouth that are causing plaque. To which I rebutted, isn't there a bacteria that causes cavities? That's a different bacteria. Okay, then I want to keep these so they can outcompete those, because I don't want those to get a foothold. So you can also have a balance between two not really helpful bacteria and choose the lesser of evils to an extent.
Yeah, or one day you should kill one and the other day you should kill the other, and then it should be fun.
Just take turns. Yeah.
Moderation in all things. Yeah. In terms of how you do your research, the technology that you use, this high throughput gene sequencing, and, you know, as your Twitter handle and all your social media, phylogenomics. Yes. Oh, yes. The phylogenomics. How is the technology today? I'd love to dig into a very specific example because people are talking about COVID-19, the coronavirus right now. How is stuff like this, the gene sequencing technology and the phylogenomics, where is it now and how is it helping us in these situations understand the microbes, the viruses that are affecting our lives and where they're coming from?
Yeah, I mean, there are sort of 3 or 4 different layers to this. I mean, the study of microbial diversity generally, and that includes, I include in microbes anything that is sort of alive that you can't see without a microscope. So viruses I count, even though some people don't count them as alive. I'm fine with calling them microbes and counting them. And bacteria and archaea, this weird other group, and single-celled eukaryotes. And For many of them, historically, it was hard to grow them in the laboratory, and it still is for many of them. And most of the way we study single microbes is by growing large amounts of them in the lab and doing experiments in the laboratory with those cultures of microbes. And that's not the only way to study microbes. And there have been a whole suite of methods that have really advanced a lot in the last 20 or so years that allow you to study microbes in their natural environments. Microscopy is the most obvious one of these, and there are some amazing microscopy methods that allow you to do real-time imaging of microbes inside, some ecosystem, and do all sorts of interesting fluorescent imaging and such things. And there's a suite of these methods that have all advanced quite a bit. The one that I use, and the one that has really been used quite a bit in studying microbes, is DNA sequencing. And basically, DNA sequencing, like with the human genome, the cost and speed has improved at faster than an exponential rate over the last 20 years, so that when I started doing DNA sequencing as an undergraduate on these symbionts of weird invertebrates that didn't have mouths and digestive systems, I sequenced a single ribosomal RNA gene, one gene from one bacterium, 1,500 letters, 1,500 base pairs of DNA. It took me 4 months or so, and probably cost my advisor $30,000 or $40,000 in reagents and other things. And now today, I have students in my lab that can sequence 10 billion bases of DNA in a day for $1,000.
Yeah.
And that change in the technology allows all sorts of things in biology to be easier than it used to be. And one of them is very important for microbes, which is it allows us to go to environments, collect samples, grind them up, extract out the DNA or RNA, and read the sequence of those. And that then allows us to figure out a lot about what types of microbes were present in those samples and make predictions about the biology of those microbes. Mm-hmm. So that's one aspect of DNA sequencing that we use a lot in my lab, is going to environments and, destroying them and extracting the DNA out of a fecal sample or a swab or a tissue sample or so on, and then using it to infer. And it works very similarly to how a paleontologist would go to like a flood site and find thousands of bones and then try and reconstruct the organisms that came to that flood site. And, sometimes it's easy. You find a bone that is a Tyrannosaurus, bone from that system. And sometimes it's really hard. You find a bone and you're like, I don't even know if this is from a vertebrate. It's weird. It's, some strange thing I've never seen before.
What is it? Yeah.
And with DNA sequencing, it's the same thing for us. Sometimes the pieces of DNA we get, we can make sense out of. If it's a well-studied ecosystem like the human gut, we can make a lot of sense out of it. If it's a poorly studied ecosystem, we can make less sense. And then the second part of the story is more what's going on with the current virus, DNA sequencing, which is that we can use analysis of the DNA sequence data to reconstruct the evolutionary history of organisms that we get samples from. Just like you would do this with humans, you can do the same thing with microbes. And that's what they're doing to try and figure out what's going on in epidemiology of various infectious diseases. There's no other way to do it with a virus. Like with, animals, you can look at their feathers and their bones and their behavior and other things and figure out something about them. But even for them, DNA sequencing is pretty powerful. But for bacteria causing an outbreak associated with lettuce or a new virus that comes in, the way to figure out its evolutionary history is to do DNA sequencing. And then just like you would compare the bone structure of a bunch of vertebrates to figure out who's related to whom, we do the same thing with analyzing DNA sequence data. And then, if it changes rapidly, Like in the course of an infection, you can even use DNA sequencing to track transmission from one individual to another, if there have changes that happen during the course of those infections.
And so you can do comparisons. Do you do comparisons between the virus that you get from one person, virus that you get from another, and you then like say, so with like coronavirus, this COVID-19, we don't know much about it. We don't know much about its mutation rate. So when we see how it changes from the bat or the pangolin or the person to person transmission, is that how we start to understand the mutational changes that when you do those comparisons along
that's certainly
its trajectory?
Yeah, that's one way to start to tease apart. If you have reference points, like you know that this sample came from this person on this day, and you know that this sample came on another day from a person they interacted with, you can back calculate, you can then count the differences in their genomes and start to calculate what the rate of change is over a course of time. You can also do that by doing other types of experiments in the laboratory by looking at, if you had a phenotype, you could screen for the phenotype. That's what people do for E. coli mutation studies frequently. You don't even have to do DNA sequencing if you have a phenotype, a color, a pattern that you can screen for. But for, many of these new infections, we do a lot of this work through DNA sequencing.
And are you using for your evolutionary relationships when you're looking at, say, the microbes in the seagrasses or in the experiments we haven't even talked about in built environments and, looking to see the relationships between these bacteria that are in the various communities that you're observing? Is that a similar method? Is that kind of what you're doing there too?
Yeah, so most of what we do in looking at communities of microbes is looking at things that are very old. So old evolutionary events that happened, millions to billions of years ago. And so we're looking there, you look for things that change slowly within the genome of an organism. So in a bacterial species, there are regions of the genome that are very constrained. So even if they get hit by a mutation, the organisms that have a mutation in that particular region of the genome die, and they don't change very rapidly. And if you're interested in old events, you want to look in regions of the genome that change slowly. And if you're interested in epidemiology, you want to look in regions of the genome that change really, really rapidly.
Yeah.
So you look in different parts of the genome and in different parts of these organisms, depending on what your question is. For most of the work we've been doing for the last, 10 or 15 years, the time scale of the questions we're working on is, when seagrass invaded the marine environment from fresh water 40 or 50 million years ago, what happened? Or when cats diversified into all the different lineages in the Felidae, what happened? We're doing things in my lab that are, a little bit older than most of the people doing population genetics or epidemiology, which is what's happening with the virus now.
Yeah, it's so similar techniques, but just different timescales and observing different parts of the genomes.
Yep.
That's okay. That's interesting. Now, I brought up the built environment work that you're doing. You've talked a bit about the seagrass. I think we all live in built environments, right? These offices, the basement that I broadcast from.
Oh, I'm in a forest right now. What are you talking about?
Yeah. What are you looking at there? I mean, that to me is this human constructed environment that we put HVAC systems in and air recycling. And what are you finding? What is going on?
So, it's funny because 10 or so years ago, the Sloan Foundation created a program in microbiology, the built environment. And when I first heard about this program, it sounded a little weird. I knew that the built environment had stuff going on that was important. And I worked on one little part of this, which was detection of biological weapons attacks, which we thought might be in the built environment. And so people were thinking, how are you going to detect anthrax in a building if you don't know what's in the building? Right. You don't know what the background is. And that was the only thing I had thought about really in terms of the built environment at the time. So I worked at this place, Tiger, the Institutes for Genomic Research, that did the DNA sequencing for the anthrax investigation by the FBI of the anthrax letters. So we were enmeshed in biodefense related questions. And then when the Sloan Foundation came along and said, we want to create a whole program in this area and we don't want it to just be about biodefense, I started to think about the questions you were just asking, which is, it turns out humans spend, in industrialized nations, humans spend like 90% of their time in human-built environments. And people have studied the microbial ecology of built environments for 100 or so years, but there hadn't been a systematic effort at a funding agency to really start to get people to address this on a larger scale. So I have books on my bookshelf back there about microbiology of the built environment from 50 years ago. There were people thinking about this, but there wasn't a lot of work on it, except for one area, which was pathogens in the built environment. So like, how long does a particular pathogen survive on a surface in the built environment? Legionnaire's disease in water, the bacteria in water coolers, and how long does MRSA survive on bed rails in a hospital? There was a lot of work on that, but there wasn't work on the rest of the ecosystems. Generally, there wasn't much. And so like, what's going on with all the other microbes when you try to kill the staphylococcus, or when you filter everything else out, what's going on with the community, or when you don't filter it, what's going on? So that it became really interesting to me that we have, you build this building, it's kind of a new semi-clean environment, and then you colonize it, and it becomes an interesting microbial ecosystem. And so, I was recruited by the program officer in this program actually to work on communications issues related to microbiology of the built environment. And I'd never done anything formally related to communication. So basically I said, I could do that, but I need to do research too, or I won't. I don't understand this communication thing.
What is this communication thing?
And that's
Hello.
So we started to do little mini projects on microbiology of the built environment to just sort of understand what was going on. And we did many of these. My favorite was we got astronauts to swab the International Space Station. It's a tiny little study because it turns out astronauts are busy, and getting astronauts to do things for you is expensive. Who would have thought? So we got 16. We got basically 16 swabs back 2 years after we thought they were going to come back from the space station. They come back in a ship that went to Kazakhstan or somewhere, and then had to get picked up and brought back to a lab, and then we did DNA sequencing from those swabs. And again, it not shocking in retrospect, but basically the space station has lots of microbes. People knew this before we did our study. There are people breathing, there are plants in the plant wing, there's lots of stuff up there. We knew that there were microbes. But what was kind of cool is that because we have so much data on Earth now, we could ask what was the space station most like compared to Earth habitats. And again, not surprisingly, it's like a home. Like
any particular room in the house?
I can't reveal that. I'm sorry.
The bathroom.
That's what I was thinking.
No,
There's a little too much going on at once.
That's what we sort of thought that it might be, maybe more like a bathroom or something, but it was just, like basically a home environment from the, the small amount of data, admittedly, that we had. And the reason I was interested in the space station is, again, this I'm always trying to figure out the rules of a system. And the space station is interesting because even though it does have people there, it doesn't have a lot of doors and windows opening to the outside world, in case you don't know.
There's no spring cleaning.
Yeah, that's right. Doesn't have many
anything transient coming in. Yeah,
there are no
outside.
Yeah, that's right. There's no dog coming in and out through the dog door. There's not many termites, at least. I don't think there are any, but and so it just seemed like an interesting ecosystem to work on because of that constraint. But again, like there's lots of really wildly interesting health related stories and energy related stories and just general biology related stories to what's going on in homes and office buildings, and as people care about now, cruise ships.
Yes.
And other hospitals are a really scary and interesting built environment in terms of microbial ecology. And when I started in this program, it felt weird because we, as mostly an outdoor ecologist, I considered the built environment fake. It's not a real ecosystem like outside. But in the end, it's a really interesting place to study ecology of microbes.
Even what was it? Years ago, we were talking about the NASA clean room. Discovered they had something that had survived all their best efforts and was eating the lead paint off of.
So we actually, because
I try not to eat lead paint. Sorry.
Because of this Sloan Foundation program, we actually are collaborating with multiple people at JPL on sequencing and analyzing the genomes of the microbes from their clean room.
Oh, wow.
Cool.
So for that reason, my past life was working on extremophiles, organisms that could survive radiation and desiccation and other things. And that is basically what's left after they try to sterilize their clean rooms. And so, there's no place on the planet that we can make completely sterile, basically. And what's left after you try to kill everything is really interesting from a biological point of view. And NASA's interested because that's what's left on the space vehicles that are going to Mars or going to some other place. So if
that's what's on the moon now.
Yeah,
that's what's on Mars now. Yeah.
Yep. So if you then discover it in the rock that you're bringing back to Earth,
right?
Don't be too surprised.
Yeah, exactly.
Hold off on that announcement until we've done some comparative analysis on this.
NASA has not been very good at holding off on those announcements. But
but it's impossible to know if it's there because we sent it there on our equipment, or if it came to us from there.
And without doing this is the point
without Jonathan's studies.
We're gonna announce. Just wait a couple days. We're gonna announce. It was nothing.
Yeah,
don't worry about it. They get a little excited with the pre announcement. Before they always get the whole story. But
that was a nightmare.
But I feel like, to Blair's point, that it studies like the ones you're doing, where if we can get that background information of what we're sending to the moon, to Mars, that if at some point we do get a hit, oh look, we found something, we can compare it to that genetic, to these genetic samples, and go, how much is it different? Is it similar? If it's similar, how similar?
And if we have. Go ahead.
I was going to say, even if we don't have a direct link to something that was found, we should still be able to tell evolutionarily that it's come from this planet, shouldn't we?
Well, but the problem is pieces of Earth have gone to Mars and pieces of Mars have come to Earth over time. And, over 4 billion years, the real question is, did anything survive during those exchanges, and did it stay? And which way did it go? And so it can be a little bit hard because I consider there probably are hundreds of millions to a billion species of microbes on the planet. And we have sequenced the genomes of, 20,000 of those. So if we find something from Mars, from the rover, and it seems a little bit out of place evolutionarily, and it's a DNA-based life form, it's clearly related to life on Earth, it's actually pretty complicated to figure out if it came from here or there, unless unless we had a sample like the work in the clean room of everything that might be going from Earth to Mars. And, you know, that is what they're trying to do. We actually have a collaboration with someone who has called this thing the Genomic Encyclopedia of Spacecraft-Associated Microbes, to build a full catalog of all of the microbes that you might find on the spacecraft, to then be able to rule them out if you find something else.
That's great, because I would assume
that acronym is interesting. It's GeoScam.
GSCAM.
GScam.
So, but wouldn't you expect, if you released microbes on Mars, an insane amount of adaptive radiation really fast? Because in theory, they're moving into this kind of new space.
They might already have it.
Yeah,
unless it's already there.
No, they might. I mean, they might already have the physics.
Yeah. It depends on what you mean by insane amount of adaptive radiation. So, like, we know from Earth that adaptive radiations happen all the time in some environments. A volcano shows up, and, you know, a new island shows up, a new host shows up in some way. And there are parts of the genomes of those organisms that go do wild things from an adaptive radiation point of view. But there are other parts that are severely constrained, that don't change very much. So that, what I would be looking for is, you know, there are parts, at least on Earth, of the genomes of many organisms that change pretty slowly, even when there's strong selective pressure for adaptive radiations.
Cool.
And that those I would expect to still reflect something to do with the time when an event happened. And you might, again, maybe on Mars, even the constraints on those things have been relaxed and weird things might happen with them. But based upon what we've seen on Earth, I think that's unlikely.
Wow.
I can't believe it's already 9 o'clock. Oh my goodness, time is flying, and we're having so much fun. Yes, you are gonna have to come back for sure.
I will come back.
Yes, to talk more microbes, because we love this. This is great conversation. But you're not leaving, are you? Would you? We have another half of the show to go, and we're going to talk about science. Jonathan, for our audience, can you let people know now where they can find you online?
Yeah, I mean, I use as my handle for all sorts of things this word, phylogenomics, PHYLO genomics. I, for better or worse, invented this word many years ago. I now rail against bad genomics words, but I'm stuck with it.
The omics. So that's right.
Phylogenomics, like evolution, philo, and genomics. And that's my Twitter handle and my Instagram handle. I have a blog, phylogenomics.blogspot.com. Phylogenomics.me is my lab website. So basically, phylogenomics and something, they can find me.
That's fantastic. And in addition to microbes, they will get wonderful pictures of birds.
Yes.
Your bird
my other obsession.
Yeah. I love it. I love it. Thank you so much for all of this microbial knowledge. I've been really appreciating this conversation. We are going to take a break. This is This Week in Science. We've got science coming at you. I've got stories about the moon. Speaking of the moon, we will be talking more about that in the second half. We will be right back with more This Week in Science. Thank you for listening to This Week in Science. You are the reason that we're able to do what we do every week, bringing you up to date and down-to-earth views on science discoveries. And with your help, we can do even more. It is true. Together, we can bring a sane perspective to a world full of misinformation. Head over to TWIS.org right now. Click on the Patreon link and choose your level of support. We have many levels. You can choose the one that fits your life the best. Be a part of bringing sanity in science to more people. Support TWIS.
And we're back. You're listening to This Week in Science.
Oh, yeah. We are back, and let's talk about the moon.
Let's.
You know that wonderful line from Star Wars, "That's no moon."
That's no moon.
That's no moon. I have been looking at this story that just came out this week about a new moon that is orbiting the Earth that researchers have discovered recently. As of February 15th, researchers, astronomers looking at the night sky, they noticed this car-sized satellite that wasn't a known satellite. And so they started checking it out and looking at it orbiting and what its pathway, its orbital pathway seemed to be, whether it was just passing by or whether it actually was in orbit. And after observing this little tiny moon, the Astronomical Union's Minor Planet Center announced on February 25th that their observations have been confirmed that this object known as 2020 CD3 is temporarily bound to the Earth. We have a new moon. There are some people who have been looking at JPL's projections of the orbits around the moon, around the Earth, and looking at the orbits, they've been saying, oh, but a couple of them appear to intersect with the Earth. And so there are people who are wondering if perhaps this is not necessarily a natural satellite, an asteroid that has been captured by the Earth's gravitational force, but maybe perhaps something that is artificial and was launched and then landed, maybe relaunched again. So is it a moon? Is it not a moon? We must do more observations.
Can a moon be a moon if it is so temporary?
And so, yeah, so that is one of the interesting things, the questions that, yeah, something that has a semi-permanent, I mean, it depends on the temporariness, but they are calling it a mini moon, calling it, it is a natural satellite around the Earth, and that is a definition of a moon, is a natural satellite.
I have a bad feeling about this. No, it's fine.
They sure it's not a giant moon that's just far away.
Right. Nope, not a giant moon that's far away. This is...
We're safe. We're okay.
Yes, it entered an orbit around the Earth in 2017, around 3 years ago. And they know this based on watching its orbit now. So based on the way that it's moving right now, they've been able to do algorithmic calculations to determine its orbital path around the planet and when it possibly was captured. And they're estimating that it will stay with us for a couple more years, maybe, but then it'll at some point bloop and it'll fly off into space. We
need to get Mariba Jah back on the show. Talk about this one.
I want it.
You want it?
Yeah, we should get it.
We should keep it.
Yeah.
Go get it.
Just put it on the UC
keep it.
Davis campus as a sculpture.
Yeah.
Our mini moon.
No Death Star.
Yeah.
It's not a Death Star. It's car-sized. It's a couple of meters across, so it's not a massive object either.
Is it a Tesla by any chance?
It might be a Tesla.
Yeah.
Yeah, but you can follow one of the astronomers on Twitter. His name is Kasper Wierschos. I will link to the story on our website. I cannot pronounce the astronomer's name, unfortunately, but he has been tweeting about his discovery. He's got some cool pictures posted up from the Catalina Sky Survey in Tucson, Arizona, and some videos that they have, some GIFs, not a GIF, a GIF, that they have put together of the movement of this new natural satellite. This is not the first time it's happened either. We apparently capture these little asteroids every once in a while. Sometimes we see it and sometimes we don't. But in this occasion, we've actually seen it.
So we have a bunch of tiny moons all the time. But like that's what bothers me is what is the difference between a moon and just junk?
Well, if it's in a stable orbit, this thing is in a fairly stable orbit right now, but it's going to, because of its size and the pathway, eventually it'll be like, I gotta go, and it'll escape gravitational bonds.
But we're not losing the big moon anytime soon.
Not anytime soon. It too, however, is pulling away from the Earth. And so at some point, we will lose the moon. And losing the moon is going to be bad, because then the tides and all sorts of stuff is good. Like, that's not good. Yeah, we need our moon. But it's not going to happen anytime soon. No.
Okay.
No, don't.
None of us will be around to
yeah, and when I say natural satellite, we shouldn't be. Yeah, I hope not. I mean,
if any of us will be, it's going to be me. You're going to have my head in a jar.
Yeah,
there you go. Oh my goodness. Natural satellite also. It's an asteroid, not something that we put up around our planet, which is an artificial satellite.
It's space garbage, not human garbage.
Yes.
Yeah.
It all depends on the perspective of the garbage. Yes. Yeah. And speaking of China, because China has been going through a lot of trouble recently, they have had a couple of science wins in my book. One of them is the Chang'e 4 mission to the far side of the moon. The Chang'e 4 spacecraft landed softly on the far side of the moon and released the U-2, number 2, which is Chinese for jade rabbit, and I'm sure I'm pronouncing it incorrectly, and I apologize for that. The U-2 rover was released and has been exploring the far side of the moon using ground-penetrating radar to figure out what is underneath that surface. What is it like? Big question. We've been to the near side of the moon. The Apollo astronauts landed. It's super dusty. The regolith is this fine dust that gets into everything. And we wanted to know, is that what the far side of the moon is like also? And the answer is yes. Yes, it is. It's
the moon all the way around.
Regolith all around. And this regolith, according to the ground penetrating radar, extends very deep. So the radar went down about 130 feet or so, and they were able to find that there were alternating layers of the really fine sand, this dusty dirt, and larger boulders and rocks. And these alternating layers, it's kind of in accordance with the idea that there was a bombardment and the moon really got the brunt of it at some point, and so dust and boulders, things shot off the surface of the moon and then fell back down again and arrayed themselves. So if you had a jar of dust and rocks and shook it, you would end up with a somewhat similar array to potentially what the surface of the moon looks like if you were to dig into it. Yes. Anyway, the rover's doing great. It's exploring the surface of the far side of the moon, and we are learning more about the moon all the time thanks to this scientific partnership with the Chinese government that is allowing us to explore a place that we have not yet been, which is very exciting. Should we talk about Chinese seaweed?
Yeah.
Yes.
Yes, we should talk about billion-year-old Chinese seaweed, and this is not something you order at a Chinese restaurant. No, this algae, fossilized algae found by paleontologists in China, publishing in the journal Nature, Ecology, and Evolution. These researchers from Virginia Tech have described a new old seaweed called Proterocladus antiquus. And they think it is the ancestor of the first land-based plants and trees on the planet. Plants and trees showed up about 450 million years ago. We have other evidence of green algae, this predecessor of green plants, going back about 800 million years. But this fossil, they found this in Dailan in northern China. They are about 2 millimeters long, very small millimeters. These fossilized strands of algae that are 1 billion years old. 1 billion years old. That's green photosynthetic algae.
Yeah.
Which pushes
they
it way
stole
back.
bacteria.
Right, so that the bacteria were there
Yeah.
first. The algae stole that ability. And then that allowed the algae to inhabit the marine ecosystem where this would have been 1 billion years ago.
And it pushes it back 200 million years from the earliest.
Yeah. There have been other discoveries of red and brown algae that are even older, but those are not as linked to the plants and the trees that we appreciate, that give us our oxygen. The seaweeds, they were important. Yes. And so the researchers were saying that there's actually a debate within the community of researchers who look at these old fossils and these old species and try and figure out what the procession of different species were in which places. And the researchers who published this study, they think that the green algae and this evidence from 1 billion years ago kind of strengthens their argument that the green algae got its start in the marine ecosystem, shallow marine ecosystem, where it was probably fairly warm in the water, but at the same time, that's where the green algae got its start, and then moved into the rivers and the streams and into terrestrial ecosystems from there. Whereas other researchers think that algae might have gotten its start in the rivers and the streams, moved out to the marine ecosystems, and then onto land. But
and then
what?
Oh, go ahead.
No, go ahead.
Oh, but then the question is, did the plants move and then the bacteria followed, or did the bacteria migrate to land first?
Yeah.
And then
so that's why we're interested in seagrasses. They're like the whales of the plant world, where their ancestors were not in the marine environment. They were in the freshwater environment, and then they moved back to the marine environment. So we called back to the sea or something like that. So their associated microbes have done a lot of, shifting between, it's 1 billion years. It's not that long.
It's the blink of an eye.
Yeah. In the life of an algae.
It's just a fraction of the, if we were put it in monetary terms, a fraction of the wealth of Bezos.
Yeah. Sure.
But yeah, I'd be really fascinated to go back and get some of those soil samples on the banks and to see if what colonized the land and soil first.
Yeah, unfortunately, these fossil algae, they were soft, and the fossils are not giving up any DNA evidence for us to look at.
1 billion years, they wouldn't be giving up DNA no matter what they were stored as.
No, it's a little old for DNA storage. As much as people want to use DNA for long-term storage, that's not going to cut it. Justin, what stories did you bring tonight?
So listeners to this show have been aware that Neanderthals and Denisovans interbred with current modern humans, and that they interbred with each other. So while Neanderthals currently are thought to have split from current modern humans about 800,000 years ago, Denisovans have been thought to have split much earlier, around the same time or as much as 500,000 years before Neanderthals did. Though there's new research is suggesting that Neanderthals and Denisovans were really the same thing when they arrived in Eurasia. These populations of Neanderthal-Denisovan or Denisotals, whatever you want to call them, eventually became separate groups that then over the next 700,000 years would occasionally interbreed, before then interbreeding with humans 50, 60,000 years ago. Although there's one event that pushes that to 200,000 years ago. There's some signal that some current modern humans snuck out of Africa a little earlier than the rest. And it would help to explain why these groups previously thought to have been separated by hundreds of thousands of years seemed to have been able to interbreed without much problem. But that's not the end of the story, because the researchers from the University of Utah delved into the genetic history data that we have so far and are suggesting that when these two groups were one, when they arrived in Eurasia. So they were, when they got here, they were one, but they weren't alone. Something was already there. Now, we kind of knew that there was something already there. A super archaic population that separated from other humans about 2 million years ago. This super archaic population, they think, based on the way they've got their analysis, had some pretty good population size, between 20 and 50,000 individuals at the time the Neanderthal-Denisovan got there. And that they interbred with that group shortly after they arrived. Now, this is then the interbred despite some isolation of hundreds and hundreds and hundreds, but even more isolation than we had thought that the Denisovans and the Neanderthals would have been. About 1 million and almost 1.5 million years of isolation. This is lead author, anthropologist Alan Rogers. We've never known about this episode of interbreeding, and we've never been able to estimate the size of the super archaic population. We're just shedding light on an interval on human evolutionary history that was previously completely dark. However, I feel like we almost covered this story a year ago. About a year ago, we talked on the show about a deep learning algorithm that looked at sections of genetic code that was only found within Asiatic peoples, and it had a sort of mystery origin. It didn't really fit Neanderthal. It didn't really fit Denisovan. The investigators from the Institute of Evolutionary Biology applied deep learning algorithms and some other in a computer model, and they came up with the source as being a Neanderthal-Denisovan hybrid. And at the time, I happened to have a winter housemate who was a physicist who I had to ask all kinds of questions about how deep learning modeling works. And basically, we talked about on the show, I was a little put off by the fact that it could only give you answers that you gave it in the first place.
Right.
So even the researcher at the time was saying that it's possible that there is also another answer to this. So it could be that this is that hint that the deep learning algorithm couldn't add information to and say can't just be these two with some. It's another, but also the difficulty of separating it because there's interbreeding with this archaic in the things that we know interbred with the current modern humans, and so. Again, braided stream of humanity, on and on. But yeah, ancient, and it's also sort of interesting because they mentioned at some point that this is the possible candidate for this archaic human was a early Homo that had left Africa. And then what's sort of interesting is that I immediately picture early Homo and Neanderthal, Denisovan interbreeding. But of course, there's the 1,300,000 years of it evolving on its own in isolation throughout a large region. So we don't really know the morphology. We can't really predict how it was evolving there in its isolation. So there is this really, really mystery archaic hominin out there that has contributed to many of our lineages.
I love this. I love that we're discovering the mystery hominid, that there was this species out there that we have yet to find evidence of beyond the the genetic interspersal, and that we don't have the bones. We don't have their story yet, but we're finding these hints that they were there.
Didn't I see one of them in the forest?
You may have.
Yeah. Right.
It turns out Bigfoot got to get a footprint of this thing first. Yeah.
Bigfoot. Oh my gosh.
Inquiring
You're
minds
not in the
wanna know,
Pacific.
how big were
The Pacific Northwest. There we go.
Yeah.
Well, no, I'm sorry. It would be a Yeti. You're right.
A Yeti.
Yeah.
Yeah, looking at this, thinking about how all of the gene flow would work. I mean, you say braided stream, and then when you start thinking, it's like a French braid. I don't know, you're pulling in one piece of hair from over here, adding it to the rest. I'm going to pull in a little over here.
But a really messy one.
A very messy French braid. Yes, It's like somewhere between,
Someone who didn't learn how to do it right.
That would be me.
And we have something that looks very archaic in the fossil record that has yet to really be identified. In terms of its relationship, because we don't have DNA yet, which is the red deer cave people who outlived the existence of Neanderthal and Denisovan. I mean, the fossil find there, I think, is only like 10 to 15,000 years old of a site with very archaic features. So it's a candidate, or there's plenty of mystery to this human story here. Also a mystery.
Hominins, when they could, they did.
Oh yes, yes.
Very true.
Also a mystery is this recent discovery. So mitochondria. We need this. Multicelled organisms cannot survive without mitochondria.
The powerhouse of the cell.
Powers the cell. Without it, cells just die. That is except for a recently discovered parasite type creature which doesn't have a mitochondrial genome. It is the first multicellular organism to be found without one. It is strange in that it lives its life in salmon and is completely free of oxygen dependency. So life started to develop the ability to metabolize oxygen, breathed some way over 1.5 billion years ago. Larger single cell something engulfed the smaller bacterium. Instead of just getting absorbed and turned to food, they formed a partnership. They made friends. The symbiotic relationship then resulted in the two organisms evolving together. Eventually, those bacteria became organelles called mitochondria that every cell in your body, except for blood, maybe, has large numbers of these mitochondria and are central to oxygen respiration processes. So they break down oxygen, produce a molecule called adenose triphosphate, which multicellular organisms use then to power the cells. So it's impossible, right?
Except it's a symbiote. It's a parasite. So obviously it's getting what it needs from its host, and it can survive these. It's devised a way to survive relatively fairly hypoxic conditions, anaerobic, no oxygen, low oxygen. But would it be able to survive the depths of space without a salmon to suck on?
But then the question is, could this thing be a lone survivor of evolution without mitochondria? A life form that evolved separately from all other life on Earth that somehow managed to evade the niche resource competition for all this time? No, actually, it turns out it's related to corals and jellyfish.
Yeah, there you go.
Jonathan waving. No, absolutely not.
Well, I had to get to my reveal quick. Because
sorry.
They used some deep sequencing of Larson's microscopy to study the Hennegaia salmonicola and found that it lost its mitochondrial genome at some point when it no longer needed it. In addition, it also lost the capacity for aerobic respiration, and almost all of the nuclear genes involved in transcribing and replicating mitochondria. Interestingly, it did keep genes that are usually associated in jellyfish, with stingers, which is what it uses to hold on to the host. And they also
it needs its host.
Yeah,
I think
like
the mitochondria left because they didn't want to be sucking on salmon.
I'm out of here.
Yeah,
I don't like fish.
I'm done.
Oh, yeah. I think this is such an interesting story. We talk a lot about this, the question of convergent evolution, when things gained traits versus losing traits. And so this is, to Justin's point about, would this possibly would this be some holdout with no mitochondria and anaerobic life form? We haven't found anything else. I mean, we're looking.
Well, so there are eukaryotes that don't have mitochondria, right? It's the multicell. Yeah. This is weird because people thought they had a rule, right? Yeah. Multicellular eukaryotes had to do this, and because they're all need this extra energy to make big organisms and to have multi-cells and things. And the cool thing about biology to me is there's always exceptions. I mean, if you say, like I told you earlier, I'm looking for rules about the microbiome, but really I'm looking for general tendencies. Because these things, always someone searches in some weird place with some weird organism and it's ditched some process that until last week people said was essential for that group of organisms.
Although you could, if you're generalizing, you could say that mitochondria started as a symbiotic relationship, and this is a parasite. So it's still utilizing a symbiotic relationship in order to be multicellular. Like, we all need somebody to help us out once in a while.
Yeah.
Some people need a salmon, I guess.
But yeah, it's the whole there was this whole debate about in my head. It's a debate in my head. Nobody else is having this conversation. About why it is that life hadn't shown up in more. Why do we don't have more than one origin story of life on the planet? And of course, we talk about niches. Everything, all the niches are filled. There's no room for it to show up again. But also, life started on an oxygen-free planet. We didn't have, well, not complete, but no oxygen in the atmosphere at least. And so we have a planet where life flourishes, which may be very different from where life can get an origin, can get a good start as well. So when we're looking for life on other planets now, we also have to remember it doesn't have to be like this one.
Yeah. Unless there are salmon there.
If there is salmon, that's a pretty good hint.
It's a salmon planet.
Things are a lot more similar than we might have thought.
Oh, I wonder if there's salmon coming up in the next segment of our show. I wonder. No salmon.
No.
Hey, if you just tuned in, you are listening to This Week in Science. And if you're interested in a TWIS shirt or mug or other item of TWIS merchandise, you can head to twis.org, click on the Zazzle link, and browse our store and buy something. But as you're doing that, it's time to listen to Blair's Animal Corner.
With Blair.
Yeah, where's my music? There it is.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have a couple of amazing studies. So I think longtime listeners to the show will know that I get very excited about certain methods behind some animal studies, and a couple this week are just so amazing. I had to bring them. So this first one is from University of Exeter, looking at seagulls. And if you think about seagulls, as I do, someone who's worked at a zoo for almost 20 years, you think about a seagull coming down out of the sky and stealing your food right out of your hands. They're very good at living near humans.
I had a seagull try to swallow my big toe once.
Ew. Well, anyway, I'm not sure what we could draw a conclusion about that from this story. Let's figure it out. But this study is looking at how humans' behavior can impact seagulls' interest in food. Herring gulls in this study showed two different buckets. So a researcher approached individual gulls in the wild, and they placed these two buckets on the ground in front of them. Each was covering a wrapped flapjack. The buckets were removed, and the researcher picked up one of the flapjacks, handled it around for 20 seconds, gave it a good look, touched it a lot, put it back down, and then they observed what happened next. Duh. In total, yeah.
I don't know. I don't want to mansplain by any means.
Uh-huh.
But for anybody born after 1930, a flapjack is also a pancake.
Yes, that's correct.
Okay.
But in University of Exeter, they were doing this in Cornish towns, so there's a little bit of a culture difference there for the verbiage as well. So then they tested 38 gulls total. 24 of them pecked at a flapjack. Great. Okay. 19 of the 24, 79%, chose the one that had been handled. So this is a small sample size preliminary study, but it does look like there's an overwhelming percentage that goes for the one that was touched by a human. To see if they were responding only to human touch, they repeated the same experiment with sponges that were cut into the same size and shape as the flapjacks, the pancakes. But in this case, the gulls had no preference for one or the other. So there was no significant change in what was touched. So it sounds like the gulls are more likely to approach food that they have seen people touch or drop or put down. So they might associate areas where there are people eating as an easy meal. I'm guessing it's more the other way, that gulls for many, many generations have known to go to where humans are eating to eat. Yeah. So they're able to recognize what is good food based on what people are touching. I don't know, potato. But this highlights the importance of the way that we dispose food, of course, near where there are seagulls, because if we're not throwing away our waste properly, then they see us touch food and put it somewhere, and that's free game for them. But so it's just it's one of those stories that I think makes sense and sounds kind of obvious, but we really had to test it to see, because you can wonder, we know the whole bird brain idea. Birds are actually pretty smart. We know that also urbanized birds are usually pretty smart because they're able to use human information to help survive. But you could see how over many generations, they would just start to recognize human food. End of story. So for this to be kind of an added element, that they'd rather eat food that was touched or interacted in some way with by a human, that kind of adds an extra layer. I don't know why they would rather eat a flapjack. I'm sure they taste and smell the same. So I'm not sure what's different about the fact that it has been touched by a human, other than that maybe they wouldn't recognize it as food otherwise. I don't know. It's very odd.
Right. I'm wondering though if they're looking at these gulls in an urban area, and I wonder if they could look at the gulls in, like at Mono Lake or something, in an area where there aren't a lot of people, where it is maybe not as urbanized and the gulls aren't used to people being around. Would they, I doubt that they would have a similar interaction, right?
Right.
Is this something that's learned? Is it something that is cultural for certain groups of gulls?
Right. Or is it related to gull culture, right? So it's not just that gulls have kind of passed through cultural information that humans identify food, but also maybe gulls interact with food and they like to steal food that other gulls have. And so
they certainly do that, right? They're very into steal. I'm a birder. I go out every day now. I try to, and then There's a section that I went to this morning with about 500 seagulls of many different species.
Oh my gosh.
And just watching it, one of them finds like a crawfish or some other thing, and as soon as it gets it, the other ones are like, oh, look at that.
Oh.
And they're starting to come over and check it out and then fight. And then, there's definitely, maybe they also do this with the crawfish, not just the flapjacks.
Right.
Yeah.
Absolutely. Yeah. So I think next we'd want a bigger sample size. We might need varied food items. We need to see if they have a slight preference for one food item over another. If that changes, if the lesser item has been touched by a human, how much does this impact actually what they're interested in?
And is it just somebody touched it first? Another gull, another person,
right?
Somebody handled it, so it's, as Kira in our YouTube chat room is saying, maybe it seems safer to them.
Mm-hmm. We're like food tasters for them, right?
Yes.
So that's, I think the other kind of interesting thing about this is I bet a lot of people who get terrorized by seagulls and perhaps don't like them very much would like to think that seagulls are just dumb and eating whatever. But this also shows a food preference, period, which I think is more than people would usually anticipate from a seagull.
My toe was not food. At least I didn't think it was food.
Did it smell like hot dogs?
Had you touched your toe recently?
Oh, that's a very good question. Had I handled my toe?
Was someone else?
You handled it, you put it back on the ground. It's free now.
For your toe. Yeah. These are all questions. So moving from food preference to stabbing speed preference.
Oh.
This is the latest
Stabbing
story
speed.
in mantis shrimp news.
Stab, stab. It's mantis shrimp news.
It's mantis shrimp news. So this is from University of Minnesota. And of course, the animal corner has begun, and so my dog has started playing with the noisiest toy in the house. Are you kidding me? Okay, so University of Minnesota, they do a bunch of studies with mantis shrimps there. And so one of the researchers there with mantis shrimp, Kate Feller, was looking at mantis shrimp for a whole different study. And the way that she did that was she would hold these mantis shrimp with their striking appendages out of the water, and their gills beneath their tail would stay submerged so that they were super healthy still and able to breathe and everything. And they were studying electrophysiology. But they found that they got really mad when you did that. They didn't like it. And she said, quote, "They have a general baseline of being angry," which I love. Relatable. And so when you do that to them, they especially don't like it, which means they're doing a lot of stabbing or smashing, depending on what kind of mantis shrimp they are. And they found that as they were doing this, there was some interesting stuff going on. So they tell it. Greg Sutton from University of Lincoln was wandering past the University of Cambridge lab bench when someone was playing with mantis shrimps and noticed the exposed. Can you please see the exposed crustaceans? And commented that it would be interesting to measure how strong the blows were in the air versus the water, because we know how strong the blows are in water. They're very, very, very strong and very, very, very fast. They create these little sonic booms underwater, right? So no one had done that before. So knowing that the animals launch them at the speed of a bullet, it seems like they could do that in air or even faster, because there's the stuff in the way. The water's thick and viscous, and that could potentially stop their speed a little bit. It could slow them down. So knowing that, they wanted to try it in air. So putting all this together, they stimulated them to strike. And that's a fancy way of saying that they poked them in the tummy. And they hated it, as one might. And so they looked, just to the naked eye, very fast. And that's great. So it seemed about the same, maybe faster, maybe slower, couldn't really tell. But then when they analyzed it, it was actually half the speed. It was half the speed of in the water. So it was actually way slowed down. And so they were trying to figure out why this was happening and really kind of confused because they found that in water, they could have actually 10 times as much power behind the punch than they were having in air as well. So it was slower and it was not as strong. So they found a previous paper looking at how they could actually, other crustaceans could kind of fine tune some of their reactions. They also found papers looking at crickets and how crickets can actually have kind of special energy absorbing structures to help them deal with potential joint damage from kind of really strong, fast reactions in air. And so the kind of hypothesis that they are now working on is that without drag in the water, and without any special appendages to help deal with the kind of the blowback of these very forceful stabs or slams, they are intentionally slowing down and softening their ability to attack or punch or stab, so that they don't blow out their very own joints in these appendages. So
that sounds like it's important to survival.
Yes.
Right?
Yeah.
Yeah, you don't want to have to be recovering from a hyperextended... Do they have elbows?
I mean, they have too many joints for them to be analogous to elbows, but something like that. An elbow-ish thing for sure. Yeah.
Yeah.
Well, and also when you have an exoskeleton, if you not only could you blow out the joint, but you could potentially rupture your exoskeleton in certain places too. So
that sounds like something to avoid.
Yes.
Yeah, I know in just boxing or kung fu, you learn very quickly never to extend your arm all the way out when you're punching. If you're punching another person, if you're punching the air, if you're punching a bag, it's one of the first things they teach you because it is a common injury for people who are just starting out, hyperextending their elbows. All sorts of those aerobic kickboxing classes, people just throw their arms out into the air and then like the next day my elbows hurt.
Yeah.
There's a reason for that.
Yeah. Mantis shrimp's been doing this forever. They know what's up.
Oh, that's right. The ladies at the kung fu, at the kickboxing class, they're no mantis shrimp.
No,
they're not. I think that's interesting. The mechanism for it and what sensors are being used by the mantis shrimp to determine the drag, the viscosity of the fluid that they're in.
I just love the idea of two different studies that were published this week. One that involved touching flapjack or pancake and then seeing if a seagull would eat it. And then the other one poking mantis shrimps in the tummy to see how fast they would try to punch them. That's animal science at its very best. And I love it.
What happens if I poke it?
Yeah.
They
what happens if I poke the pancake?
They should poke the seagulls in the belly.
Yes. Oh my goodness.
They'll eat your toe.
You really got it for the toe.
I do. Okay, it is time for our quick science stories at the end of the show. It is International Polar Bear Day tomorrow.
Ooh.
And as International Polar Bear Day is coming upon us, reports of cannibalism among polar bears are increasing, and Russian scientists reported these cases of cannibalism, which have been long established fact, but the researcher Ilya Mordvintsev was quoted as saying that the cannibalism is increasing, and it could have to do with lack of food, reduction in sea ice, but it could also be partly due to more people working in the Arctic and being able to report the behavior. So we don't know whether it's a case of reporting ability or actual climate change and food availability changing, or a little bit of both?
So my audio feed cut out at a very specific moment in what you were describing, so that I don't know what's doing the cannibalism, but the fact that there's more people, and that might have been, it made me think that this is the Inuits who are doing this. And I know that can't be right.
No, polar bears.
Polar bears. Thank you very much.
Polar bears.
Thank you very much. Oh my goodness.
And these cases of cannibalism are usually larger male polar bears cannibalizing the cubs of females because they're defenseless aside from the defense of the mama bear. But this is usually the way it goes down. But it's a little sad. So I hope we can find a little joy for our polar bears tomorrow and do whatever we can to help sustain the environments that they live in, conserve the places they live.
And as draconian as that sounds, we see that in lots of places in nature. Apes will do this. Lions will do this.
Sure.
The act of, and it is a form of it's a form of self-population control, which obviously has worked because these organisms are surviving.
Yeah, but now the polar bears their numbers are declining.
We're the pressure. We're the pressure and selection that's causing it in this instance.
Just a quick item about it, and that is that male polar bears in particular are always or often will eat, especially polar bear cubs if they're in their territory. Which is why it's so important for a female to find a good area to den, and also why it's so important for polar bears to have giant territories.
A lot of space.
And so with the sea ice forming later and later,
Right.
there's less and less space, and these animals are forced to have overlapping territories that did not before, and that's causing some of these problems. If you can do anything tomorrow for polar bears, just talk to somebody about climate change, something they can do in their neighborhood for climate change, maybe about a community choice aggregate, have them sign up for green energy nearby, and just tell somebody a cool polar bear fact. How about that? Everyone can do that.
Ask them not to go to the North Pole or anywhere near it. Just don't go. That's an option.
That's one option. Moving on from polar bears, a study published in the Proceedings of the National Academy of Sciences looked at the picture of gender equality in academic publishing and studied the number of women in academia over the past 60 years. The study ended with researchers whose careers ended in 2010. The study started in about 1955, and they found that although men and women publish a comparable number of papers per year and have equivalent career-wise impact for the same total number of populations, there are different publishing career rates, and they find that women are dropping out of academia at all stages of the career pipeline. So a lot of the effort has been recently to maintain the junior level female faculty in academic departments, where that is a wonderful place for effort to be continued. The picture coming from this story suggests that it may be reworked to try and maintain women along the academic pipeline throughout careers. So a very important study to figure out what is going on and why do the women drop out of academia? We don't know that yet, but although numbers have been increasing since the 1950s, there are still differences in publication rates as a result of dropout. And then finally, I have some poems for you.
You wrote me a poem? How sweet.
I did not, but I have some poems for you. This is called Sadness Model Poem. A long trail of falling mist had made its way here, and now airily it seemed as if to drown the discordant thunder clang. It seemed to drown the music of the rain in this lost place of sorrow, far off.
Nice. A little extra wordy.
Did it make you sad? Did it evoke sadness?
Yeah, I
yeah,
kind of felt
yeah.
like they should have done a little bit more revision. It's sad that they they committed to that final version. But other than that, you know.
All right, how about the joy model poem?
Ooh.
Heard I a song of joy, a song of happy sound, fills all the air I breathe. To him I sing the happy song. All night long on the steep green grass I ride and sing.
Okay,
I like
like
the
that
first one better, actually.
No, I like the second one better. Oh, by far.
I think that pretending to be deep with sadness is easier, or you can get away with more nonsense, maybe, because you can be like, oh, they're depressed. Like, oh, there's figure.
I think we have a problem here, which is the words are only part of the story. I think you may have read the poems at slightly different styles.
I did. You're right.
Okay.
I did. So I may have imbued the poems.
Yeah.
With my emotion. Researchers from the University of Colorado at UC Colorado Springs, they have published in the archive a preprint of their work with machine learning, in which they gave their algorithm, they gave it texts from the, where did these things come from? They came, I'm looking for the exact places that they trained. Oh, from older Gutenberg poetry, from the Gutenberg database is what they use. They browsed through the Gutenberg database from Project Gutenberg and UC Santa Cruz's DreamBank databases. So they gathered these texts from these two different databases of language and words, and they used an emotion lexicon data set called Emolex that's been developed by the National Research Council of Canada. And they split their databases into emotion categories, trained up their algorithm to write poems. And they said that for the most part, it did seem to produce poems that elicited sadness or joy a significant amount of time. They also had these algorithms, these computers, do dream poetry. So it's more dreamlike poetry verses.
Very
I
cool.
am in the air. I have another poem. I have a dream poem for you right here.
All right.
I am in the air. A little bird in my cradle comes to me and says to me, "Oh, what a beautiful little bird is this that I have made. I am a little bird in my cradle. I know the bird. I have known it. I have seen it when I can't see it."
This is what happens when you give computers edibles.
Yeah.
Yeah. And also
I hated that.
Seems...
That was yucky. That was like scary.
No, it was also slightly confused about the direction that reproduction works. I don't know. There's these two things. One makes, I don't know. Just throw them both at the wall.
What is wrong with my brain that nonsense creeped me out so bad?
Yeah.
So
I have to say, the other two that you did before, if it wasn't for the title, I think I would have believed you if you were like, "This is by E.E. Cummings." But the fact that it had that very analytical title made it kind of feel sterilized. But without it, yeah, I feel like it. But this one sounds like either a robot or someone who seriously needs to talk with somebody.
Or somebody who is very young and has not yet had a lot of training in poetry and literary arts.
I don't know. There's something about it that feels like unhinged. I don't know why. That one really bothered me. I'm going to be thinking about it when I go to bed later.
Ooh, thinking of computer dream poetry.
You should have Siri read it to you.
No! At least my Siri is an Australian man. I really don't need so much. But Siri original, that would be pretty creepy.
So I had to look it up to see the name, but there's a short film called Sunspring that was created by Oscar Sharp, who we got to meet at the last Santa Fe Institute get together, who had an AI algorithm write a script. And then he just followed that script and filmed it, like all the dialogue and everything. And it's really, it's really interesting. We could be getting to that point where, most of media is a formulaic thing anyway. let's just let let's get rid of the writers and let the robots do when they compiled all of the things that people said they liked in art and ended up with this horrific landscape with like George Washington and a deer, because some people like animals, some people like and it was like this horrible cacophony of items that were thrown into this thing, but based on what everybody would like.
Yeah, the robots have not yet taken over our arts. They're getting there. It's getting closer. Machine learning is getting us there for sure, and it's examples like this that bring to light where machine learning, natural language processing is going to end up taking artificial intelligence eventually. And I find it fascinating. I mean, where does artificial intelligence end and humanity begin at some point in the future?
Well, we began already. We already started.
We began. I should have said it the other. Where does humanity end and artificial intelligence begin? Right? I'll get it right. Okay. Who wants to tell another story?
Well, right from robots dominating the arts, I would like to move on to robots with pasta fingers dominating deep sea marine science. So marine biologists have adopted soft robotic linguini fingers as tools to conduct their undersea research. Yes, this is from a February 24th edition of Current Biology. Science found that jellyfish held by ultra-soft robotic fingers shaped like linguini expressed significantly fewer stress-related genes than when they were grabbed by traditional submersible grippers, which are like giant claws. So they kind of pull, and I'm wondering if Kiki might be able to pull up the video because it's pretty funny looking, but they pull them in very gently, not very invasive. And they were able to see, like, yes, it looks more comfortable, but how do we know for sure that the jellies aren't as bothered? And they actually used genomics. So they confirmed that the soft robots were kinder based on looking at their genomics. So basically, their example is, imagine you're sitting very happily at your desk and I take a measurement of what genes are active. Then I poke you with a claw hand. I then look at how differently your genes reacted compared to when you were sitting unbothered. The strength of that difference can act as an indicator of your level of stress. This is from senior author David Gruber, professor of biology, City University of New York. So they had the jellies gently held by the linguini fingers, and then jellies that were clawed. They checked their repair genes that were expressed and indicated that they were priming themselves for physical harm. With the linguini fingers, that did not happen. So they were able to show that that method caused less stress. And so it does prove their hypothesis that soft robots are a good, effective tool for delicate interactions. And I love this. They think that this could also be applied to things on land, like harvesting fruits without bruising them, rehabilitating muscles of stroke patients, and many other rigid-bodied things that rigid-bodied robots just can't do because they're too kind of clunky and strong. So there you go.
Clunky.
Yeah, I love this. I wouldn't mind if there was like a glove or something, or that I could send to my doctor ahead of the next physical. Fantastic.
Yes, absolutely. Yes. Or just be massaged to sleep, like the head massager. Just imagine linguini fingers.
Ew.
No? I mean, they wouldn't be wet.
No, get off my head.
Would be wet like they just got off the submersible.
If you attach them to a keyboard, they could type poetry.
Oh,
that's right.
Yes, it's just kind of dragging their linguini fingers across the keyboard.
Yeah, exactly.
Linguini poetry. Oh my goodness. Any more stories?
I think we did it.
I think we did it. Yes, we have done it. We've come to the end of another show. Thank you for listening. Jonathan, thank you for joining us tonight.
Thank you for having me.
Really appreciated having you here. It was super fun. Everyone out there, I hope you enjoyed the show. And if you did, share it with a friend. Shout outs go to Fada for his help with our show notes and social media, Gord for manning the chat room, Identity4 for recording the audio for the show, and I would like to thank the Burroughs Welcome Fund and our Patreon sponsors for their generous support. Thank you to Paul Disney, Stu Pollock, Ed Dyer, Andrew Swanson, Craig Landon, Ed Philip Shane, Ken Hayes, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Richard Porter, Mark Mazzaros, Paul Calder, Jack, Matthew Litwin, Jason Roberts, Bill Cade, Eric Knapp, Richard Brian Condren, Dave Neighbor, Ashley Doyle, Maddie Perrin, Ashley Doyle, you're new, Ben Bignell, Justin Taylor, P.S., Josiah Zahner, Howard Tan, Donald Mundus, Sarah Forfar, Dan K., Matt Bass, Darwin Handon, Patrick Pecoraro, Gene Tellier, John Gridley, Corinne Benton, Adel Majoy, Sarah Chavis, Rodney Lewis, Tiffany Boyd, Mountain Sloth, Seth O'Gradney, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Noodles, Kevin Reardon, Christoph Zucknurek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Liesl Suzuki, Jim Drapeau, Greg Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for all of your support on Patreon. For information, visit patreon.com/thisweekinscience. And on next week's show, we will be back again next week.
On Wednesday at 8 p.m. Broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Want to listen to us as a podcast? Just search for This Week in Science wherever you listen to your podcasts. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've learned here today, show notes and links to stories will be available on our website, www.twis.org, twis.org, and you can also sign up for our newsletter.
Hey, got something to say to us directly? You can email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email. It'll be spam filtered into oblivion.
If you want to contact us indirectly and in public, you can hit us up on the Twitter, where we are at TwistScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And oh, it'll you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
And we have come to the end of our show. And now we're in the after show. Ta-da! There we are. Jonathan, thank you so much again. I love your microbe chain. Have you collected them all?
I mostly, my kids have stolen most of the ones that I had. So these are the ones they didn't want or that I stole back.
Right, the ones that are left.
Yeah.
That's fantastic. Yeah, I love those little microbe toys. When they came out, I was just like, yes, this is the most brilliant stuffed toy ever.
The babies are good too. They have mini versions of many of them that come in a petri dish.
Oh, that's
those
fun.
are the ones that I have. I have I have chicken pox and I have rabies.
Yeah, they're pretty cool.
I saw that in the chat, our private chat, Blair, and I was very confused.
Yeah.
Blair was in our private chat saying, I have chicken pox and rabies. And I was like, what happened in San Francisco that I missed? Yes. But thank you so much for staying late with us and doing the rest of the show.
Yeah, Jonathan, that was great. We didn't even get to touch on a bunch of stuff.
Yeah,
that you're involved in. So you'll have to come back at some point.
I would be happy to.
Yay!
I think this is a seagull feather, actually.
Is that your golf? I don't. Oh, wrong side.
Did you put that in the freezer for a week or no?
No.
Feather mites in your hair now.
No, it's been in my office for about 13 years, so I don't think there are any mites left on it.
Yeah, fair. I know.
More likely dust mites.
Whenever kids at the zoo pick up random feathers, I'm always like, okay, put that in a plastic bag, put it in your freezer at home.
Yeah.
Mites.
Oh dear. Oh.
They might be a problem.
I have a seagull friend that comes and visit. I'm on the 7th floor here. So it's I have one that comes and kind of looks in the window, checks out, what are you doing? You're on the couch again? Okay. I got stuff to do. And then he leaves. Or
the gulls. Yeah, we have gull crow battles here in the morning.
Geez.
Oh.
Yeah, they take over. It's like
crows, always.
There's the east side crows and the west side gulls, and then they meet in the middle and they battle around us. That's right. Total West Side Story.
When you're a gull, you're a gull.
You're a
all
gull.
All the way from your first poke in the belly to your
first big
to your flapjack every day.
That's fantastic. Identity 4 says it was cool meeting you at AAAS. Yes.
Yeah.
Identity 4 we saw in person at AAAS, and you sat with us in the wine bar.
Oh, excellent.
Yeah. We had pizza and wine.
Nice.
It was great. Yeah.
Yeah, that was a fun event. I was sad that it had to end. I really I kept wanting to go to and see more stuff, but then stuff would happen at the same thing. Other cool stuff was going on at the same time. It was so hard to choose where to be for those 4 days.
Jonathan, did you have a good time at AAAS?
Well, I am ending my time on the AAAS Council.
Oh.
So I had to go to,
meetings.
But I got up early every morning and went birding, which was awesome. I went with a friend of mine, Carl Bergstrom, and then I went a couple times just walking around, or I took an Uber out to Discovery Park one day. And I discovered, relevant to the previous conversation, the single most complicated thing to identify in the Seattle area are all the crazy seagulls and the hybrids that occur between all the seagulls. And I
bet a lot.
I post all my pictures to iNaturalist, and I posted a bunch of them there, and there was lots of conversation by other people saying, I don't know, that looks like an Olympia, so-and-so hybrid because of this. Do you have another picture of it? And with a different angle and different, it was.
You're like, yes, I photographed this one seagull for an hour.
Yeah, I did. I tagged it and followed it.
Nice.
And gulls are hard generally, but I guess around Seattle, there's a ridiculous amount of hybridization between species too. So they're even more complicated.
That's interesting.
I just identify them when I try to as the bigger category, which is large white-headed gulls.
Right.
And then there's like 9 species in the Seattle area of those, and hybrids of all of them. So, but it was still fun. I saw lots of cool things, and I got out. The birding thing I do more just to force myself to get outside.
That's
smart. Great.
Yeah.
That's so important, and there's so many studies that keep coming out about people needing to get outside into nature.
Yep.
And how and you're talking about how we spend 90% of our time in the built environment.
Yeah.
Yeah. So that's
awesome.
And
this is something that me and Blair have talked about a bit too, which is that on one hand, I'm advocating for people to stay completely away from nature because any interactions that we have are just terrible. But of course, then Blair points out,
that the more that contact you have with nature, the better decisions you make ecologically. Is that the one?
Exactly right.
Yeah.
So it's this really tough thing of you really need to be out in nature to appreciate it, to know what is being the reasons things need to be conserved, and to have that affinity to the real world.
But that's why you have spaces where humans are supposed to go and spaces where they aren't. That's why you have trails. That's why you have natural areas, wildlife preserves, and then you have like desolation wilderness, and you have all of these kind of different levels, so that you can kind of make sure that.
Yes, and I think Davis does a decent job with that conserved causeway space.
I post from there daily.
Yeah, there's a space where you can kind of go and, drive and then walk a bit into this sort of preserved wetland on the edge of it. And then there's this whole large swaths that if the birds don't like you, they still have plenty of territory they can stay.
It's the YOLO bypass wildlife area, and it's a really interesting collaboration between farmers and the city and some of the towns and hunters and conservationists. So they flood some areas during the migration to encourage the waterfowl and other birds that are migrating to have some habitat. But during some of the rest of the year, a lot of it is used as rice farm. It's got a hunting area, and then it's got an area that's, like you're saying, completely closed to anybody but the docents. And it's amazing. It's really interesting. And I love the sort of interface where it's just a couple of miles from Sacramento, but it's this really spectacular wildlife area. So I've been going out there every day.
Oh, nice. And there's times the migration period where the sky is full of birds. It just becomes an amazing location.
Flyway.
Yeah,
it is a flyway.
Millions of snow geese at sometimes, and it really is one of the stopping areas for the migration.
And there's something about driving out, and just especially in the winter, if it's rained, and seeing the bypass. It goes from the fields during the dry season to just this body of water, and the birds in it. It's just such a neat space.
Well, every few years when we get a lot of rain and the Sacramento River is pretty full, they spill over the water into the bypass is like a flood control zone.
Right.
And so every few years, it's the YOLO ocean, basically. It's like 10 miles wide and 20 miles long flooded zone. And it's wild. It gets, white caps, and it gets.
I know.
Yeah, and it used to be an area that would then flood into farmland south of Davis. There's a farmer's field down there that shows what life used to be like, because there's what do you call it, like a metal barn structure out there that is on about 12 feet of raised earth. And it's like this, it's right in the middle of this. And then that's where, because the guy who originally was farming there lost all of his equipment. All of his tractors and everything got flooded and couldn't recover. So when he went back, he was determined not going to change plots, but he built a place for him to park all of his equipment on high ground for that flood season.
Yeah, well, it's better. I mean, there's this area where it's called Frank's Tract on the Sacramento Delta River, and I used to sail on the Delta growing up and through high school and college. And in this area, Frank's Tract, at one point, like, it used to be farms and houses, and they decided to open the levees and to change the flow of the water. And there are places that are mapped out that you're not supposed to go to because there's like trees and tractors and things still underwater. Yeah. They just flooded the entire...
Just a couple of feet under the water, yeah.
Yeah, it's all underwater, just sitting there now. Just still been there, yeah. You just hope that your boat keel doesn't run into anything when you're on low tide. Yeah, but that's a neat area. Yeah, one or the other. Build higher or just end up underwater.
Yeah.
And it's more than birds. It's got bats too.
The bats are great. They're not back quite yet because they migrate, but in the late spring, it's not quite like Austin, Texas, but we have a population of bats that comes out at sunset every night, and it's like 200,000 bats that fly out in a stream from the bridge between covering the causeway. It's pretty amazing.
That's so cool. I used to
More
love the
cool.
bats.
More cool.
More
The
cool?
peregrine falcons come out every night to hunt the bats.
Yeah.
And it's unbelievably spectacular when they're out there, because they're just, diving at 100 to 200 miles per hour into a big, giant cluster of bats.
Wow.
Yep. I want to watch that. I need to come back to this.
We had a lab meeting out there.
That's awesome. Let's watch the bats. Let's watch the falcons.
Yeah.
That's a good. It's more of a bonding meeting.
Yeah.
Do an activity together.
Life is more important than science anyway.
Mm-hmm.
Science
Keep it
is
going.
life. Life is science.
Yeah.
Yeah. There's science outside the lab. Well, you're like, no. Free time. Well, we don't want to keep you all night long. We just keep talking. Justin, are you are you past your jet lag?
Oh, you know what? I had actually just recovered from jet lag, and then I prepping the story, which for me was yesterday. I fell asleep. So I woke up very early. I'm back. I don't know. I think I'm on sometime in the mid-Atlantic right now. I'm not quite in Denmark time, and I'm not quite
sure where you are.
Yeah.
You're in international waters.
Somewhere. Yeah, that's where maybe somewhere in Greenland. Actually, I might be on the eastern edge of Greenland right now in terms of my personal time zone. But yeah. So no, I'm not right yet. I was close. I got close.
Almost there. I almost got it. Yeah. When do you come back? Are you coming back?
I'll be. I'll be back in next week.
Next week.
This time next week, I will be on a plane, heading back to Cali for.
Wait, are you going to be here for Wednesday's episode next week?
Oh yeah, yes. So I will do the show. Yeah.
Because
so
I won't.
Because Blair's not gonna be. I mean, I'm searching for a replacement.
Saw. Can you not use the word replacement?
That's
okay. So actually, you know what? I'm glad
Stunt double
is good. Yeah.
Stunt double. There we go. That's my.
I'm glad you said that, because I just looked at the time. I might have to do a quick hour and be out to make it to my 2 trains to the airplanes to be there early enough. Yeah.
All right. I am definitely finding myself a co-host for next week.
Yeah.
Justin, just let me know if you feel like if you feel like you're gonna be needing to get
Jonathan,
out.
what are you doing this time next week?
I'm actually looking at my calendar right now.
It had occurred to me that I don't. So the flight itself isn't till like 9 something, but it's not. I just looked and it's
you have
it's
to
already
travel time
7:30, and I got to be there. I got to be there now.
Yeah.
Basically. Oh boy.
Mm-hmm.
Yeah.
Let me know, Kiki. I'm around.
Well, there you go.
Wow.
I can easily
Glad
be
we talked.
a replacement.
Oh no. And Justin and
Substitute.
Blair were never asked back again.
Substitute.
I would have my substitutes. Oh yeah, maybe I should find. I'll find people who will put we'll put your heads on popsicle sticks. They'll wear Justin and Blair masks.
I'll be on the road for about 3 weeks, and then I'll be back here again.
Little monkeys. There we go. That's perfect.
Oh, yeah, there we
Sock monkeys.
So this is actually the first episode that we were broadcasting with video. I was not quite ready to be in front of the camera. And so I had, I think, some kind of a squash or something that had a mouse sock puppet over it.
Awesome.
That represented me for maybe the first 2 or 3 episodes.
Yep. It's back there. I remember. Yeah.
Perfectly okay with being on the air as a disembodied voice.
Yeah.
But the additional pressure at first was a little much.
My office is filled with all the things I'm not allowed to put out at home. So
that's slowly happening to me too. Yeah, a lot of fun things at work.
It's all the fun things, the things you're not allowed to have at home, and the things that you want to keep safe from your children.
Yeah, many things. They're all part.
Or
of
just
the
I have the 7,000 piece Millennium Falcon made out of Legos at home. I have the 200 piece or whatever it is one at work. So it's
Yeah.
I like it. Yeah, keep the 7,000 piece one at home for sure.
Yeah.
For sure. Okay, so for everyone out there, Blair and Justin will not be joining us next week.
In 2 weeks. I'm gonna go pilot a Millennium Falcon. Speaking of,
I'll be here for the intro. I'll be here for the first half of the show. Then
no, you're not. You're gonna.
Yeah, why wouldn't I?
Because you're just gonna be. I wouldn't be. I'd be like, I gotta pack, gotta make sure all my things, and then I gotta get on the train. I gotta make sure I'm early, and because
you know how many times I've done this recently?
No.
I've many.
Well, this is your first time, right?
Huh? Oh yeah. That's right. Inside joke, but yeah, this is technically the first time I've done it no more.
Anywho. All right.
Okay. We'll see my co-host Blair in 2 weeks. Justin, keep me updated, but I imagine I'll see you in 2 weeks also.
Yeah, if anybody's looking for me in the city of Davis, I will be at the 3 Mile Brewing Company's facilities.
Nice.
You'll just be living there for a couple of weeks.
I'll just be hanging out there most days and every Sunday. Can't miss trivia. I can't miss the trivia. That's really the only reason I'm going back.
Back to Davis. Going back
not
to
for
Cali
the children,
for
just,
trivia.
Yeah, no, they'll be there too. That's also be nice. Yes.
Yeah, I want to get back to Davis. We need to figure out our celebration.
Yeah, let's do it in Davis. That sounds perfect.
I think it would be fun.
On the air at KDVS Live?
Yeah.
I can train the seagulls to like flapjack by then.
Yeah.
Yes, that'll be perfect. Look, they will eat my flapjacks. Okay. Justin, what's the word that you say?
Oh, say goodnight, Blair.
Good night, Blair. Say good morning, Justin.
Good morning, Justin. Good
night,
Kiki.
Good night, Jonathan. Thank you so much for joining us.
You're welcome. Thanks for having me.
Oh, it was fantastic. Absolutely. I really am sorry that we waited so long to make this happen, and can't wait to make it happen. Maybe next week. We'll see.
Whatever.
Yeah. And good night, Justin, Blair, and good night, everyone out there, or good morning, wherever you are. I will see you next week. Faux show. Okay. I'll see you on the Twitters. Bye.
Bye.
And you're going to hit the other end, Brad. Yeah, there you go.